In our last chapter for this unit, we're going to talk about modern understanding of genetics, the concept of linked genes, and so on. So in the early 20th century, the development of embryos was linked to chromosomes. Later, Sutton observed that chromosomes separate during meiosis and realized that chromosomes hold the key to the process of inheritance of the traits. So essentially, the physical carriers of Mendelian inheritance units are chromosomes. As you may remember, homologous chromosomes independently arrange themselves among gametes during meiosis, so that sorting is completely random. You can also remember that gametes are haploid, which means they contain the half of the chromosome numbers of the parent sets. Now, male and female gametes have the same chromosomal number. In case of humans, it's going to be 23, which means they both equally contributing in terms of the genetic composition of the offspring. When these two, male and female gametes, fuse during fertilization, the number of the chromosomes in offspring is restored to diploid. Now, initially, there was no linkage of chromosomes and traits. But first of all, the behavior of chromosomes during meiosis fully confirmed and corroborated with the Mendel's laws of inheritance. And first person to observe the independent assortment of chromosomes was Eleanor Carothers, saw it in grasshoppers. But the final contribution to the linkage between the chromosomes and inheritance of the genes and traits was provided by the experiments by Thomas Hunt Morgan, which he did in fruit flies, Drosophila melanogaster. Now, our conversation before was circling around the concept that each trait is inherited independently. However, it may not always be true, as we now know since we mentioned the concept of linkage. So Mendel suggested that traits are inherited independently. Later, Morgan found that there is one-to-one correspondence between the segregating trait and the X chromosome, which confirmed the random chromosome segregation. However, Morgan also demonstrated that the genes present on the same chromosome may not necessarily be inherited together. However, they can become unlinked through meiosis. And I want to stop right here and reinforce what I just said. When you have two genes on the same chromosome, they may or may not be inherited together. There is a certain probability that they will be inherited together, and there is a certain probability that they will be inherited separately. The proximity of these genes and the degree of linkage depends on the relative probabilities of the genes being inherited together or not. So, when genes are linked, they would not follow the independent assortment of genes as it was predicted by Mendel. Now, the number of traits is much more than the number of chromosomes, which can be explained by multiple linkages between the genes. Now, you may remember that during meiosis, chiasmatas are formed along the chromosomes when two non-sister chromatids exchange segments. When the segments are exchanged, the alleles that are located in the segments that are exchanged become unlinked from the rest of the alleles. This crossing over results in the exchange of genetic material during synapsis, which leads to the recombination between the non-sister chromatids and the production of new combinations of alleles, i.e. non-parental types. So the take-home message from this is that when you have two genes on the same chromosome, they either be inherited as the parental phenotype, meaning that they are linked, or they can be inherited after the recombination. I apologize for that mishap, and let's move on and talk about genetic linkages. So imagine that you have two linked genes, A and B, that are on the same chromosomes. So in this example that you see on the right, the genes B plus and VG are linked. So if they are linked, then the probability of forming gametes that are either homozygous dominant or homozygous recessive are 1 and 1. If they are unlinked, the probability of having not homozygous, sorry. So the probability of having two dominant alleles in the same gamete or two recessive alleles in the same gamete is 1 to 1. If genes are unlinked, then the probabilities of 4 different gametes is the same. So if genes are linked, this is essentially a parental phenotype. Non-parental phenotypes are gametes like dominant A, recessive B, and vice versa. These gametes result from homologous recombination or crossing over. So Morgan, in his experiment, crossed the Drosophila that were heterozygous for each gene in one parent and homozygous recessive for each gene in another parent. And what he found, which was quite interesting, he found the predominantly parental genotypes and some, minimally, non-parental genotypes. Genotypes that are the result of recombination. So what does that mean? It means that the most offspring, shown here, showed linked inheritance. Some offspring showed the recombination, the non-parental genotypes. So it demonstrated, first of all, genetic linkage. Second, that genetic linkage can be disrupted via the recombination events. So in Morgan's experiments, he worked on two traits: the body color, B plus versus B, and the wing shape, normal versus vestigial. So when these traits did not recombine, so for instance, he was doing a test cross, okay, and if you look at this, he was doing the test cross and he found that most of the offspring exhibited parental genotype either heterozygous for both genes or homozygous recessive for both genes, but there was a portion of offspring that changed their genotype from the expected linkage. So Morgan's experiment essentially supported the concept of crossing over and the genetic linkage. Now, based on the results of these crosses, one can establish what's called a genetic map. If chiasmata are formed, they suggest that there is a homologous recombination. At the time, it was not fully understood how chromosomes can break and rejoin during recombination, but then at some point, Alfred Sturtevant built first genetic maps based on the frequencies of recombination, basically drawing those frequencies from the data produced by Morgan. Recombination frequency essentially tells the scientist the order of the genes on the chromosome and the relative distance between them. The distance between the genes is measured in centimorgans. It is not an actual length unit, it's a unit for gene mapping, where 1 centimorgan corresponds to 1% recombination frequency. So if the frequency of recombination is high, it means that genes are far apart. Think of it this way. If the genes are located on opposite ends of the chromatid, chances that the gene on one end will be recombined while the gene on another end will not are pretty high. If the recombination frequency is low and genes are inherited together, these genes are close. For instance, the body color and the wing size genes on the chromosome 2 of Drosophila melanogaster had 17% chance of recombination, which means there were 17 centimorgans apart. Now, if you look at this, 17% of the offspring are recombinant. That essentially refers to that idea of the recombination frequency. Now, remember that genes are sequentially linear on the chromosomes, and that recombination, crossing over, can occur anywhere on the chromosome with equal likelihood. Now, that means that distant genes, as we just discussed, are more likely to recombine because they are separated by the larger regions. So, for instance, if you cross an organism that is heterozygous for 2 genes that are supposed to be linked, and an organism that is homozygous recessive for those same 2 genes, and the recombination frequency is 5%, it means that the distance between them is 5 centimorgans, meaning that genes are linked, but not extremely close. Now, this map is just an example of the genetic map based on the recombination frequencies in Drosophila melanogaster. So, for instance, here you can see that the genes for red eyes and normal wings are pretty close. Now, the perfect linkage means that 2 genes are always inherited together, which means a recombination frequency is 0. So there is no crossing over. On the other hand, the recombination frequency of 0.5 means that genes are on the different chromosomes. It's a 50% chance that these 2 genes will be inherited together. You can do the math of the probabilities on yourself. Another opportunity is that these genes are far apart on the same chromosome, but in ideal case scenario, the genes have to be on the different chromosomes. However, when the distances between the genes become closer to 0.5, the predictions of whether these two genes are located on the same chromosome or on two different ones become less and less accurate. Because if you think about this, if the recombination frequency it gets closer to 0.5, there are two options. These genes can be far apart on the same chromosome. Or maybe our statistics aren't that precise, so we can't tell apart whether the genes are on the same chromosome and recombine super frequently, or they are on the different chromosomes. So the crossover was demonstrated in corn plants by Barbara McClintock and Harriet Crichton. Later, Kurt Stern showed that there is a homologous recombination crossing over in Drosophila melanogaster. So he crossed flies with unusual X chromosomes and recorded the chromosomal exchanges in the offspring. So essentially, what is a homologous recombination? Homologous recombination, just to understand, is the process in which chromosomes break and rejoin in precise locations. Now, since we're talking about chromosomes, we need to address the question how we identify them. The study of the chromosomes is called cytogenetics. Now, the study, essentially the determination of how they look, is critical for identifying chromosomal abnormalities. A couple of terms that you need to know. Karyotype. Karyotype refers to the full set of chromosomes in an individual. It is characterized by the chromosomal number, appearance, length, banding, and the position of the centromeres. Karyotype is reflected by the karyogram, which is the chart created by photographing chromosomes under the electron microscope. Chromosomes can be divided in autosomes and sex chromosomes. Autosomes are first 22 chromosomes in humans, and they are arranged in order of size. Sex chromosomes are X and Y. On the illustration here, there are two X chromosomes. Now, despite the fact that chromosome 21 is smaller than the chromosome 22, it is still referred as the chromosome 21 because it was discovered in the context of Down syndrome. Now, chromosomes have arms, short and long. So short arm is referred to as P, petite, and the long arm is referred to Q because Q follows P alphabetically. One day somebody will write a book on how scientists name things. Now, the locations of genes on the chromosomes are described by the designation of the arm and numerical subdivision. So let's talk about the disorders associated with abnormal chromosomal number. So these disorders can arise either through duplication or loss of chromosomes. Alternatively, it can be associated with the changes in the number of complete sets of chromosomes. They are very easily identified through karyograms and usually result from non-disjunction. So non-disjunction is when two homologous chromosomes, in case of meiosis 1, or sister chromatids, in case of meiosis 2, to separate. Now, what can result? What can lead to this? There are two main contributing factors. Either the synapsis is misaligned, or the spindle apparatus doesn't work as it is supposed to. The likelihood of non-disjunction actually increases as parents get older. So let's take a look at non-disjunction meiosis 1. So in this case, when there is no disjunction in meiosis 1, homologous chromosomes fail to separate. So at the end of the day, you have two gametes lacking the chromosome and two gametes that have an extra copy of the same chromosome. So basically, if we call these chromosomes short and long, these two gametes have only one short chromosome, but these two gametes have one short and two long chromosomes. Non-disjunction in meiosis 2 arises from when sister chromatids do not separate. That leads to one gamete not having a chromosome, two gametes having the normal set, and one gamete having an extra copy of one of the chromosomes. So abnormal number of chromosomes is called aneuploidy. Euploid refers to the normal chromosomal number. So in humans, that would be 22 pairs of autosomes and one pair of sex chromosomes. Some categories of aneuploidy include monosomy, when one chromosome is missing, or trisomy, having an extra chromosome. Usually, when an organism misses one autosome, this results in the failure to develop, essentially death, because some essential genes are lost. This balance of gene dosage between all the autosomes is crucial for the survival. In case of trisomy, there is an extra chromosome which results in the excessive production of proteins or whatever gene product is, which will lead to developmental issues. Most trisomies that are autosomal fail to develop. However, there are some trisomies of smaller chromosomes that can result in viable offspring. The most famous of them is Down syndrome, or trisomy 21. This trisomy results in short stature, short digits, broad skull, large tongue, and significant cognitive development delays. You can see the photograph of a child with the Down syndrome right here. Now, it's worth mentioning that it's been shown that likelihood of trisomy 21 significantly increases with a maternal age almost exponentially. Another chromosomal abnormality is polyploidy, which refers to the organisms with more than two sets of chromosomes. So, for instance, if there is a diploid egg that is fertilized by a haploid sperm, it will lead to a triploid zygote. It doesn't happen often in animals. There are some examples in all the way up to some reptiles, but not birds or mammals, so what we consider to be higher animals. However, polyploid animals are often sterile because meiosis does not proceed normally. So the daughter cells that are produced during meiosis are aneuploid. In asexual reproduction, there are cases of haplodiploidy, when the egg that is unfertilized mitotically divides to produce offspring. Again, not common in animals, but polyploidy is pretty frequent in plants. Plants that are polyploid are usually larger and more robust. As you can see on this illustration, when orange daylily is triploid on this photograph, and it's large and robust, and flowers have three times the number of petals compared to diploid. And this is polyploid Endelian, which is, quite frankly, a bit terrifying. Now, non-disjunction can occur not only in autosomes, but only in sex chromosomes. But humans tolerate these variations much better, and we're going to figure out why. So in females, one X chromosome in each cell is inactivated early in the development, forming bar body. Now, why is this? Because if you have two X chromosomes, you're going to produce twice the amount of gene products. So in order to maintain the normal number of gene products compared to males, you want to inactivate one X chromosome. The X chromosome that is inactivated is selected randomly. It can be paternal or maternal. And once it is inactivated, all the cell descendants, all other daughter cells, will have the same inactive X chromosome. So a good example of this is tortoiseshell cats. So they have a color variation because of the random inactivation of X. Only one X chromosome is active in each cell, but those X chromosomes can be slightly different in terms of the alleles. Now, X chromosomal abnormalities usually cause mild disorders associated with cognitive abilities and physical abilities, and usually sterility. So, absence of X chromosome results in the ablation of uterine development, so it doesn't lead to any viable offspring. Now, triplo X condition are phenotypically female, but they usually have developmental delays and reduced fertility. Klinefelter syndrome, shown here, phenotypically male. However, the testis is small, the breasts are enlarged, body hair is reduced, and there is a possible intellectual disability. Now, many conditions may result in multiple X chromosomes, up to 5. All but one X chromosome undergo inactivation. Now, in Turner syndrome, when there is only one X chromosome, these are phenotypically female. They all have short stature, webbed neck, often impairments in terms of the hearing and heart function, and they are sterile. Now we're going to talk about some other chromosomal abnormalities, and we're going to start with duplications and deletions. So in duplication, a segment of a chromosome is copied and confused to the same chromosome, or can hang out in the nucleus. In case of a deletion, the chromosomal segment is lost, which you can imagine leads to abnormalities. An example of such an abnormality is Cri-du-Chat syndrome, in which most of the short arm of the chromosome 5 is, well, deleted. Developmental cognitive issues, characteristic high-pitched cry in newborns, and overall abnormalities in the physical and cognitive development are associated with Cri-du-Chat syndrome. Now, another possible cause of deletions is pairing with homologs for gene alignment. When chromosomes in meiosis pair with homologous chromosomes and the alignment is improper during recombination, some gametes can become aneuploid for certain genes. Inversion is another example of chromosomal abnormalities. In case of chromosomal inversions, the segment of a chromosome rotates 180 degrees. It can be mechanical shear on the chromosome, transposable elements, which we don't have time to cover, but imagine fragments of the chromosome jumping around. Now, inversions usually alter the orientation of the genes, but usually do not disrupt the gene sequences. They can result in the changes in expression of the genes. If the regulators of the genes are misaligned, for instance, silencers, enhancers, and promoters, which may lead to abnormal gene products. So inversions can be categorized in peri and paracentric. Pericentric inversion involves the centromere and occurs around the centromere, changing the length of chromosomal arms. In paracentric inversion, it happens outside of the centromere, and chromosomal arm lengths do not change. Now, another interesting situation with inversion can occur in heterozygotes. When one homologous chromosomes has the inversion and another one doesn't. So during meiosis, the homologous chromosomes align, but in order to align, inverted chromosome is to make a loop. That can result to the imprecise synapses, stretch the chromosome, cause alignment issues, and ultimately lead to aneuploidy. And finally, duplications, deletions, inversions, and translocations. So what is the translocation? During the translocation, a fragment of the chromosome breaks off and attaches to a non-homologous chromosome. Benign translocation may have no effect on the function of the genes. In deleterious translocations, you can observe some severe effects. All of it depends on how the placements of the genes during translocations disrupt the regulatory sequences. There are specific translocations that are associated with cancers, for instance with chronic myelogenous leukemia, when translocation results in overexpression of the genes stimulating the abnormal cell growth. They're also associated with psychiatric conditions like schizophrenia. Now, in reciprocal translocation, the two chromosomal segments of two different non-homologous chromosomes are exchanged. In this case, the genetic material is not lost, it is not gained. All the genetic material remains just on the different chromosomes. But even such a reciprocal translocation can lead to problems, because this translocation can disrupt, as we mentioned before, the regulation of the gene function. In our next lecture, we're going to talk about evolution.